Well, good morning, folks. It's me, your old pal, Dr. Harry, Dr. McKay, whatever I want to call myself, back again with Neuro3301. So here we are. It's the day after the quiz, or the next class after the quiz. We'll talk about that next week. But yeah, so today we're going to be talking about obesity and energy balance. I want to go over a few aspects of course business as we kind of turn the corner into the last little stretch of class. I'm just trying to, sorry, I'm not really thinking too well this morning, so I'm having a little trouble getting getting my words out. Let me just jump into the lecture and start talking rather than trying to vamp off the cuff here. So yeah, as I was saying, the lecture for today is going to be obesity and energy balance. Not exactly what I would call something to do with mental health, strictly speaking, but something to do with the brain, pretty clearly. And also happens to be my own particular research focus, so I decided to make a lecture about it. We'll get into that. I wanted to talk first about, well, I guess maybe like sort of a broader checking under the hood of the course. Actually, hold on a second here. I just have to print something or get a PDF ready. I'm just going to put you on standby for a minute. Sorry about that, folks. Yeah, it's if I feel like this in the morning, I can only imagine how you all feel. Cassandra asks, did the lecture conk out for everyone else? I don't know. My stream still looks good over on my end. I did briefly pause the lecture to go and grab my notes, but I'm back now, so hopefully everyone can see me. Anyway, so what I wanted to talk about before getting into the class was just sort of an overall vibe check on the course as a whole. I noticed, I guess, maybe looking at YouTube view statistics too much gives you brain poisoning. You think about all those people like, obsessing over being silenced or canceled or shadow banned or whatever on Twitter. Looking at the stats too much will do this to you. And I don't think this is really, I think from what I've heard from other instructors, this is a fairly common phenomenon having to do with the overall like vibe level of the university as a whole. But I thought it was kind of interesting because given that I can download the view data, I can look at the performance of my lectures and see sort of how students are reacting to them and how the class as a whole is going. Now, so what I made here is a cumulative plot of lecture views, where the y-axis is the cumulative views. So every time somebody watches a video, the number goes up a little bit. The date, of course, is here along the x-axis. The first midterm is given by this vertical line, and the second midterm is given by this vertical line. The individual lectures are represented by each individual curve, and they're labeled here. And the number of students in the class is given by this horizontal line. So in principle, once a lecture has had more views than there are numbers of students in the class, on average, every student has watched the lecture one time. So you can see a very interesting effect as the class goes on, where the first lecture almost immediately had, more views than there were students. So the first lecture was quite widely attended, and by the time the first midterm came along, it had been watched over 150 times. And the same kind of rate of growth was visible for all of the other midterms in the first. Oh no. Sorry. Thank you for chatting with me. I was just ranting and ranting to an empty slide. Oy vey. Okay, so this is, sorry folks, this is not it's not going well this morning. We all have mornings like this. But anyway, we're back. So what I was saying was the fun of the live stream. You know, you could just turn it off and do it over again, but we're live. We're live. This is the fun of live TV. Anyway, so what I was saying is that the y-axis of this plot represents cumulative views. The date is on the x-axis, and the view rate of all the individual lectures are plotted by these individual lines. So as I was saying, like by the time the first midterm came along, that's represented by this vertical line, the first few lectures had been viewed more times than there are students in this class, which is about the amount of times you'd want them to be viewed. But a very interesting pattern emerges for the second set of lectures, which is that the view counts tend to be fairly low, and in the last couple of days leading up to the exam, they climb up, and by the time the second midterm is held, the lectures have been viewed on average once per student. So you can see there's a very interesting contrast. Alexis asks, how many students are there in the class? I think there's like 68 or 69 altogether. Pretty close to 70. So, and of course, I don't know who's watching, so I can't say on a student-by-student basis who's watching, but it is interesting. I mean, you know, you'd imagine most people would watch it probably once and then move on with their lives. So it's hard to say. But anyway, I think it's a very interesting pattern to see the new kind of modality for following the lectures is to sort of leave them to the last minute and watch them. Now, I know that doesn't include anyone here who's watching live. And I know that like this, I'm not, you know, trying to make a suggestion that this has anything to do with your success on the midterm, but I think it's more of an interesting pattern that we're observing across the school and across the department of just kind of a malaise in the air. It becomes very difficult, I think, for students to stay motivated to keep up with lectures. So this is something that many people are observing. I happen to be able to like plot the data out in a pretty convincing way, but I think this is something that's observed quite widely across the department. I think there are a lot of factors involved. Student engagement, I think, is at record lows across the school. Online learning, especially the sort relying on voluntary adherence to a schedule, tends to be problematic, I think. It's a bit like the promise of social media in general. I think when Facebook first came out, people were promised that this was going to bring everyone together and it was going to make socializing so much more easier. But now we're all sitting in our basements watching our aunts and uncles post QAnon memes. And clearly something went wrong there with the way the social media delivered. Coming to lectures is a bit like eating vegetables at dinner. Not really too pleasant, but probably necessary. Anyway, so I would say if you're looking for something to kind of change in the last third of the course, what I would recommend is trying to watch the lectures earlier, as close to as possible on time, so that they have some time to percolate in your brain. Generally speaking, the course is difficult enough without having to look at the very last minute for all of the content. But I sort of recognize that things are not really normal or too enjoyable for people these days. Again, across the board, and this is by no means an easy class. So I don't think that anyone can be blamed for how they react to the material or how they manage their time. But I think it's kind of an interesting fact about online learning. I mean, if I had been offered online teaching as an undergraduate, I probably would have gone for it, but then I also probably would have been diagnosed with depression and dropped out after, 1 or 2 semesters. Myself, I'm pretty introverted, but I think when it comes to introversion, you can't get high on your own supply. You have to kind of go out there a little bit. But anyway, there's not really much we can do about this. Anyway, my thinking is that for the last exam, I'm going to I don't know what the marks for the midterm are yet, but I think what I'll probably do is come up with some kind of scheme to help you to maybe reallocate some of your marks to sort of incentivize really putting a lot of effort into the final exam. I haven't figured this out yet, what I'll do precisely, because I need to see the marks for the midterms. But it would probably be something like I'll let you, I'll let you drop your lowest midterm if your mark on the final would improve things. It'll be some kind of scheme that allows you to try to get a really good mark on the final exam and then boost the mark overall for the course over, disproportionately to how much the final is worth. So I think I'll do something like that to try to incentivize learning and take a look at the content for the final exam as we get closer to it. I don't want to talk about the midterm too much because I really truly don't know anything about it. It hasn't been graded yet. The machine graded parts actually looked pretty good. So the parts that were graded like multiple choice, that were graded automatically, those actually looked quite nice. So I think that people hopefully did okay on it, but we'll have to wait and see. Anyway, so more details on that next week. I just thought, I'm sorry to ramble about all this view statistic stuff. I just think like, I don't have this data for everyone else in the university, but I have to assume that this is pretty typical of where people are at, or where students are at in their experience. I think that online learning is very, very tough for people. I think it's very tough to be motivated to come to class or to do the lectures or to do really anything. I can completely sympathize because I know myself and I know how I react to that type of unstructured environment. So it's just neat to see it play out in real life. And there's only so much I can do to incentivize you to change your course, but we'll try to work something out when it comes to the final exam to make it worth your while to really put all of your try to study ahead of time and do a really good job on it. And then more grades. So we have the marks back for assignment 2 for everyone who we have submissions for. The average here was a little bit lower than the first one, 82.38. The max score was 98 and the minimum was 61.5. So thankfully nobody failed the assignment. I think most people did actually quite well on it. The median is 82.69. So that means half the people got better than 82.7%, which I think is quite a nice mark for people. It sort of has this like nice positive skew that we see for a lot of the online assignments. They tend to be sort of people tend to do better on them than the exams because you have kind of a lot of time. So the third assignment is coming. The TAs are just working on their test run of it today, or not today, but this week. So we should have that posted up hopefully by the beginning of next week, and you can get started on that. And I think the third assignment is maybe a little bit simpler than the second one. So hopefully that'll work well for everybody. Okay, so enough course admin. Your old pal Harry, he just loves looking at data. And I think everything can be understood as a data question. So if looking at the class grades or whatever informs me about better ways to structure the class or better ways to sort of teach or incentivize learning, then that's always what I'm going to try to do. Okay, so let's get to the actual content here. Why obesity? So obesity is a dimension of human phenotypic variation. It's considered to be sort of a pathological state. If your body mass index is above 30, body mass index is really just a ratio of your weight to your height. And it's just sort of a simplified way of indicating whether somebody is like heavy for their height. So it's actually kind of a bad measure in many ways because you can be heavy for your height even if you're not, even if that extra weight isn't made up of fat. It could be muscle or it could be bone, or you could have different kind of anthropomorphic characteristics that would make your BMI higher than it should be or higher than normal, but you're still not considered to be like obese clinically due to your body fat. However, on a grand population-wide scale, it's reasonably good at estimating this kind of thing. The rate of obesity has been rising worldwide in recent decades, mainly since the 70s. The rates of obesity can be quite high in Canada. So this is a figure from Maclean's magazine showing the rate of obesity across the country. The darkest colored provinces have the highest rates. So Newfoundland has the highest rate of obesity. The Maritimes in general tend to be a little on the higher end, as are the prairie provinces. BC has the lowest, and Ontario has the same as the national rate, which is not surprising because almost everyone in Canada lives in Ontario. This is something that I remember having to explain to people when I was living in the U.S. They didn't know very much about Canada, but it's like, Canada is like consists mostly of the people who live in this kind of corner of Ontario. Almost everybody, statistically in Canada actually lives below the 49th parallel. So like below the kind of canonical U.S. border. So that has a kind of a biasing effect on the national statistics. Obesity is quite closely tied to socioeconomic status in rich nations. In rich nations, it tends to be the case that low socioeconomic status, poverty, low education level, all kinds of other elements of SES, they're all associated with higher rates of obesity. So people with less money and less time tend to have higher rates of obesity in rich nations. Now, in poor nations, the situation is reversed. But in rich nations, there's not exactly a shortage of food. There's really more a shortage of good food for people in a certain sort of segment of the population. The reason we care about obesity and body weight variation in general is not so much for obesity itself. It's more the correlation between obesity and a range of poor health outcomes like cardiovascular disease, type 2 diabetes, and neurodegenerative diseases. I think this is something that, from my own perspective, and maybe you'll kind of catch this as I go through the lecture, I think that perhaps too much is made of obesity per se. It's very, very tough to change your body weight in a lasting way. I will try to explain why this is in this lecture. It's very, very, very difficult, and to the point where you're almost asking sort of too much from the average person to persistently change your body weight if their natural set point happens to be, kind of toward the overweight or obese side of the spectrum. Much better, I think, to focus on the actual health outcomes themselves and try to live the healthiest life you can for the body you happen to have. This isn't to say that you can't endeavor to lose weight if it's needed medically or if you'd like to for whatever reason. It's just that focusing strictly on obesity and body weight itself is the wrong approach, I think, for society, because it's just so difficult to change, and it's also just such a source of stigma that it really doesn't, it doesn't help anybody to look at things that way. However, this is the way that the data have been defined for us. This is the way the phenotype has been defined, and this is where all the studies are based. So even if we don't really like the terminology or like the kind of medical perspective, then we're still sort of stuck. We're still stuck using it. So the condition of obesity has long been assumed to be quite heavily genetic. If you look at twin studies and adoption studies, they find that the heritability of obesity is between 40 to 70%. And that would make it a fairly highly genetic kind of condition. Now, the genetics of obesity, they're a little bit mysterious, as are most things. We've been through this story before. We have a condition with rather high heritability, but when you go looking for the genes, they're nowhere to be found, or at least they're very elusive. The genetics of obesity, unlike some of the other conditions, we do have a divide between what I would call monogenic and polygenic categories. There are seemingly a fairly large number of monogenic causes of obesity where a single gene is mutated in some way that, massively increases your particular risk of obesity or your body weight being above average. So this is something that seems a bit more common with obesity than other conditions. It's difficult to say why precisely this happens to be the case, other than the fact that, perhaps many genes ultimately contribute to the control of energy balance. Energy balance, by the way, is just the balance between energy in and energy out in your body. Energy in is what you eat and what you consume, and energy out is whatever you either don't absorb or burn off in some way, whether it's resting energy expenditure or whether it's exercise or whether it's thermic effect of food or what have you. So ultimately, your body weight is determined in some sense by your energy balance, because if you eat more than you burn, you will gain weight, and if you burn more than you eat, you will lose weight. And if things are kept constant and you eat exactly as much as you need every day and burn off every calorie that you eat, you'll maintain a stable weight. And it's probably telling that most of us actually do maintain a fairly stable weight. Over the years, we'll gain weight gradually as we age, but for the most part, we gain very little weight, meaning that as uncontrolled as our eating and activity levels seem to be, they're actually held in fairly tight balance by our brains. Our brains are able to sort of behind the scenes turn little dials to change your metabolism or change your activity level or change your appetite in such a way as to maintain your energy balance at a fairly neutral point. But anyway, it may be the case that the maintenance of energy balance is something that involves many genes. And if you have a lot of genes that all contribute in their own kind of important way to a given phenotype, then it seems like there would be a higher likelihood of there being monogenic disruptions. Because if you have all kinds of genes to choose from, all you would need is one to be disrupted for the whole system to break down. Compare and contrast, I suppose, with a very highly refined system where there's only a few genes involved, the odds of any one of those genes mutating is lower. You know, it's a bit like it's a bit like why is it I'm trying to think here. Well, why is it easier to break a computer than it is to break an old-fashioned telephone? Well, there's more parts in a computer, so there's more things that can go wrong. So you might take from this the idea that the regulation of energy balance is one of the main things our genes and our brains are concerned with, and there's many places where there can be problems. However, one thing that both the monogenic and polygenic causes of obesity seem to converge on is this dependence on brain and neurodevelopment related genes. This suggests that obesity is caused by the brain, and that is the central thesis of my talk for you today. And I think this is really something to keep in mind. I mean, here we are in neuroscience. We have some people in the neuroscience department who are very focused on this kind of question. So maybe it's not quite as new to you as it might be to somebody else that hasn't been in the neuro department at Carleton. But it really is quite astonishing to think that the basis of obesity is most likely the brain. It's not the fat, it's not the muscles, it's not the skeleton, it's not the pancreas, it's not the digestive system, it's not the heart, it's not the lungs, it's not the kidneys. The brain. The brain is where all the action is when it comes to body weight regulation and obesity. And I think that's just a little bit surprising to people because obviously when you are looking at the natural variation in body shape and size that exists in the population, probably the first thing you would think is there must be something going on with the fat or the muscles or what have you. The brain is maybe not the place you'd think. But the brain is where, you can't eat without a brain, and you also can't move without a brain or burn energy without a brain telling your cells what to do. So at some level, the brain does make sense. So we can compare and contrast the varieties of monogenic and polygenic causes of obesity. This has sort of the same kind of it follows the same pattern as other as contrasting monogenic and polygenic causes, say of schizophrenia or neurodevelopmental disorders or something like that. Monogenic have a high genetic contribution. Involve a single mutation in one gene. That mutation has a large effect. They tend to be comparatively rare. Monogenic disorders are not common because they would be sort of not favored by natural selection, so they would never reach a high level of allele frequency. Penetrance means they have high penetrance, meaning that if you happen to have the mutation, it will have a very strong effect. There's not really very much recessive alleles going on. And then environmental influences are minimal because the allele is so penetrant that it doesn't really matter what's going on in your environment. So we got a question from Nathalie. I'm just reflecting on the previous slide, but is there a reason why the Maritimes and Prairies have the highest obesity rates in the country? I would have expected Ontario to be the highest. Yeah, I think that I can't say for sure, to be honest, but we do see that the Prairies have fairly high rates. Newfoundland really has the highest, as do the Maritimes, but the Prairies are the next highest after that. And I think a lot of it would have to do with the physical infrastructure, rural versus urban. How much do you drive? How much do you walk? These kinds of comparisons. It would have to do with the overall socioeconomic health of the region. The Maritimes tend to be one of the more poor regions of Canada. A lot more people are poor and working class in those areas, and just by sort of the relationship between socioeconomic status and your ability to find food that's healthy, to afford food that's healthy, to have time to cook it, etc. Those things all contribute to obesity, so they kind of cluster with the provinces as well. When it comes to the Prairies, I don't. It's hard to say. I don't really know what the Prairies are like. I've never been there, but it's pretty similar in the U.S. actually. The Great Plains and kind of the upper Midwest states tend to have pretty high rates of obesity as well. And I think in the U.S., Colorado has the lowest, and Colorado is kind of a little bit like BC, I guess, suppose. But yeah, I don't know. It's a variety of factors. It's kind of tough to say just looking at the map without getting too far into speculation, into speculative territory. And then Alexis asks, could you repeat the definition of penetrant, please? Sure. So penetrant just means that if you have this allele, it will have a strong effect. So a gene that has an allele that has very low penetrance is one that would be sort of like a recessive allele, right? If you have the bad version of it, it may not do anything to you if it's compensated for by other genes you have or some other kind of environmental characteristic, some other environmental factor. But if it's highly penetrant, if you happen to have that allele, then it will have an effect and it'll have a fairly strong one. So these are the kind of the properties of monogenic causes of obesity. They're also early onset and they tend to be fairly severe. The polygenic ones have a modest genetic contribution. They involve hundreds of genes. Each variant in each gene has a small effect. They tend to be fairly common variants with low penetrance, and they rely heavily on the environment as a determiner of your actual risk factor. So you're talking about gene-environment interactions there. So I want to kind of take a bit of a diversion because we don't really have the time or, I guess, space to stretch out and talk about every possible monogenic and polygenic variant of obesity. So I thought I would maybe just give you a little vignette of one famous case, and we can kind of talk about that a little bit, and then maybe that will serve as a template for a broader understanding of this if you ever want to stretch out and look at it more closely in your own time. And maybe you know this story. If you've taken any of Alfie's classes, this might be something that you're familiar with. But I think it's a pretty interesting story, so I'll tell it all the same. Back in the 1950s, scientists at the Jackson Lab, and the Jackson Lab is a big mouse breeding facility in, I think, Bar Harbor, where scientists breed mice for laboratory use. And if you breed enough mice, like if you just continually breed mice, you'll eventually find some that have rather interesting mutations because, mutations happen at a certain rate. And if you just keep breeding them, you'll eventually see one. So they discovered a mouse that had a spontaneous mutation that produced massive obesity. So this is the fat mouse here. His weight is actually more than the weight of 2 regular mice combined. So they're, on the basis of kind of classical genetics and breeding studies, they determined that this obesity-producing mutation was due to a mutation that the fat mice were homozygous for. So they were called OB mice. The gene was just named OB because it was before the genomics era when they could go and map the genes. They just said, okay, there's some hypothetical gene called OB, really an allele of the gene, and if you have this allele, you're obese. So these mice have 2 copies of this OB allele, and people knew that genes coded for proteins, but nobody had a clue what proteins these genes coded for. And there's a couple of thoughts you could have. Maybe they code for something in the fat cells, maybe they code for an enzyme, or maybe they code for a hormone. Hard to say, right? So in the 60s, our old pal Douglas Coleman, who was working in endocrinology at the time, came up with a rather clever test. And the basic idea of the test was if OB codes for a hormone, then it's probably something that, it's probably a hormone that they're producing wrongly, or there's probably a problem with whatever hormone it codes for in the obese mice. So if the mutant mice are lacking a hormone and wild-type mice happen to produce this hormone, then what you could potentially do is transfuse blood from a wild-type mouse to a mutant mouse. And if the wild-type's blood contained some kind of vital hormone or vital essence, then it should reverse the effects of the mutation. Now, rather than go there and continually transfuse blood, what you could do instead is parabiose them. The model is called parabiosis, and para just meaning beside and biosis meaning life. So what you're doing here is you're essentially surgically joining the circulatory systems of the OB mouse and the wild-type mouse, creating sort of an artificially conjoined twin. The result looks like this. These mice have been given, these are differently, the fur color is different on these guys, but typically that wouldn't be the case, just to show you how it looks. And what this allows is for hormones from one mouse to travel to the other and vice versa. And it turns out that OB mice that were parabiotically joined with the wild-type mice lost weight and became healthier. And that suggested quite clearly that whatever OB is, whatever this gene does, it codes for a hormone of some kind. So there is a circulating satiety factor, as Coleman would have called it, and people were very curious about this because, it's even back in the 60s, people were very curious about body weight and obesity. And if you found that if you found evidence, I suppose, that there's some kind of hormone floating around that if you give it to somebody who's overweight, it makes them skinny. Imagine the value of that, not only from a health perspective, but also from a commercializability perspective. Imagine what you could do. So people were on the hunt for this for quite a long time. But in the 60s, there was only so much that could be done when it came to genetics. You could certainly argue that a gene existed on the basis of selective breeding experiments and that kind of thing. But to actually find where the gene is and identify what it codes for is considerably more difficult. So we had to wait all the way until 1994 when our friend Jeffrey Friedman came along. He's this guy here, and identified the OB gene and the protein it codes for. So he discovered that this OB locus is located in a gene that codes for something called leptin. He named it leptin from the Greek word meaning thin, leptos. Leptin is secreted in proportion to fat stores, so the more fat you have, the more leptin that you produce. And so in this way, it sort of acts as a negative feedback regulator of feeding. So as your fat stores increase and as your need for food goes down, the level of leptin goes up as well and makes you less hungry. In principle, that's how it should work. And so mice with the OB mutation don't produce leptin, and thus the satiety promoting nature of leptin is lost in these mice. Now, this of course immediately suggests that mutations in leptin might be responsible for some cases of human obesity. I mean, early on, I think once people had made the recombinant protein, there was a dream that it could be the panacea for all obesity, and whoever was able to commercialize it would make billions of dollars selling, this magic diet hormone. It turns out that that's not really the way it works, but we'll see. So there are actually cases of human monogenic obesity that are centered around leptin. So here, this was Montague et al. In 1997. Once this mutation was first discovered in mice, it was pretty quickly mapped over to humans, and it became possible then to screen human obesity for mutations to this leptin gene. So here is a pedigree from a human family from the Montague et al. Paper in 1997. And the people who have this severe monogenic obesity are colored in black. The squares are males and the circles are females. And their genotypes are given by the little letters underneath their names. So N means they have a normal leptin gene, and M means they have a mutation. So you can see that if you're homozygous for the mutation, you have this severe obesity. If you're heterozygous, as this person is and as the parents happen to be, then you're fine. This is a highly consanguineous family. Consanguineous means basically it's sort of a nice way of saying incestuous, I think. So you can see that like these great grandparents gave birth to a number of kids, and then those kids had babies, and then the cousins got married. So not a great setup, really, especially if you have monogenic forms of disease floating around in your gene pool. Because you can see how, like, if you're heterozygous for this fairly rare mutation, you're probably going to be fine if you're just breeding with a random person off the street. But if you're breeding with somebody who's in your own family, then the odds of you both having this mutation are considerably higher, and thus the odds of your children being homozygous for it, they go up as well. So anyway, that's just a fun fact about this family. The mutation they have is shared. I think there's a variety of different monogenic leptin mutations, but this is the one that they discovered, and this was a fairly major finding in its day. This is a chromatogram from Sanger sequencing. Sanger sequencing, if you recall, is the old-fashioned version of sequencing that was responsible for the first human genome project and was used quite widely in the earlier days of genomics. What we are seeing here is there is a highlighted cytosine here in the leptin DNA sequence that's present in the wild types, but in the mutants, it's gone. This ACA trinucleotide thing is here, and it's still here. So actually, the sequence upstream of this missing cytosine is all the same, but in both of the obese cases, they're missing this cytosine. So what this is a frame shift mutation. As you probably know, the way that mRNAs and the way that genes are coded is with codons. And codons are sets of 3 nucleotides that specify a particular amino acid. So when the ribosome is coming along reading the mRNA, it goes one codon at a time and it says, okay, TCA, this codes for whatever, leucine. I don't know what the actual codons are, but each trinucleotide codes for a certain amino acid. And so the ability to assemble the protein properly depends on the codons being properly read in the right order. If you take out one letter, all of the codons downstream of that get shifted over by one, and that completely changes their content and their meaning. So take, for example, here, say you're starting from, I'm just trying to improvise an example here, but say you're starting from CAG, and the next codon is GAC, and the next codon is ACC, maybe, whatever. That's the correct amino acid sequence. Here, you're starting at CAG, then you get GAC, and then you get, wait, I'm doing this wrong. I'm not showing it right. Right, because this is the thing, what would actually be happening is you'd be starting from CCA. So anyway, the point is that if you take out one letter, it completely mixes everything up. This happens in coding sequences of genes, and it's one of the worst kinds of mutations you can have, because you're not just changing the identity of one amino acid, you're changing the identity of every amino acid downstream of that frame shift. So a very bad situation. And it turns out that if you give these kids recombinant leptin, it helps their obesity enormously. You just replace this one missing hormone, and their problems are more or less solved. Not quite as simple as that, but it makes a huge difference. However, despite the hopefulness of these early findings, obesity, broadly speaking, is not monogenic. When they discovered leptin in the first place, it was briefly heralded as a potential solution to obesity. And while it is for anybody who has a mutation to their leptin gene, most people don't have that mutation, and so leptin supplementation really doesn't do a whole lot for them. In fact, what they actually have so much, somebody who's overweight or obese and it doesn't have this mutation actually has huge amounts of leptin because they have huge amounts of adipose tissue. So giving leptin doesn't really help that type of person very much. It does help if you're not producing any leptin, and that's the reason you're overweight. But if you have leptin, then giving more really doesn't make a whole lot of difference. But anyway, now thanks to exome sequencing and the technologies we have available, many more monogenic causes of obesity have been discovered. So what's interesting about these is that most of them seem to be expressed in the hypothalamic energy balance circuitry. This is a part of the brain in the hypothalamus that controls your energy balance. And genes like MC4R, the melanocortin receptor, PCSK1, POMC, and SEMA3A are all ones that have come up in recent studies. Some of these, like MC4R and POMC, are directly involved in peptide signaling. SEMA3A is actually sort of a neurotrophic factor that guides the development of axons in the developing nervous system. And it turns out that that's fairly important for the development of this circuitry as well. So for most people, energy balance and obesity is a polygenic trait. Now, if it's polygenic, there's one thing we can do to find the genes, and that's a classic GWAS study. The genome-wide association studies are the ones that we're always hearing about now. The GWAS studies of human obesity have been ongoing for quite a while now, for the last decade or so. And the latest meta-analysis consists of over 700,000 people. The Manhattan plot I'm showing is actually quite old. It's from 2010, from Speliotes et al. In 2010. That's because the newest studies, for whatever reason, didn't include a Manhattan plot to show you, and it's just kind of not very fun if I can't show you the Manhattan plot, because we all have gotten really used to it. The plot here shows that one gene stands up above all of them. It's FTO, located on the 16th chromosome. I've mentioned this before offhand, and I believe I asked you a question about it on one of the assignments. It's kind of an interesting gene because it was not, it was a case of a very high, a gene with a very high effect size, relatively speaking, that nobody ever really heard about and nobody had ever even implicated with obesity before. So once it was discovered back in sort of 2007, I believe, it's spawned a whole flurry of research into what the heck it does. And there's a lot to be said about that we're not going to be able to get into, but it's quite an interesting story. Other genes would be like MC4R. Again, this is part of the melanocortin signaling pathway in the hypothalamus. BDNF, brain-derived neurotrophic factor. You have neurexins. You have nerve growth factors. POMC is over here. Anyway, pretty much all of these genes have something to do with neurons or the brain. And this bears out not just by our kind of eyeballing of the data, but also bears out statistically. Oh, Alexis just asked a question. I'm going to go to the chat window for a second here. Speaking of lockdown, do you think that obesity rates have increased more dramatically over the last 2 years since the quarantines began? I don't know, to be honest. I think that's something that we'll find out as time goes on. The epidemiology of this takes a little while to calculate. You have to do a lot of surveys and gather a lot of data. I know it's been very difficult, surprisingly, to figure out if rates of mental illness have gone up or down over quarantine, because it's very tough to say what the baseline data are, and to kind of develop a survey that would be able to show that rates of depression have gone up. As far as we can tell, they certainly have, but the exact amount that they've increased by and in whom the increase is being felt the most is a tough question. But when it comes to obesity, yeah, I certainly would imagine that the lockdowns and whatnot would be not promoting the best health of people. I remember when they first started, actually, and all the gyms shut down, there were just lineups around the block at like all the like fitness stores that sell like exercise machines and whatnot, because all these gym people were really desperate to be able to exercise at home. So I guess maybe some people got a little bit healthier, but I would say on average everyone's health has declined. Probably that will be a lasting effect of the lockdown. I think when all is said and done and you really crunch the numbers, you're going to see that the kind of quarantine measures had a lot of negative health effects that had nothing to do with COVID, just the sort of negative outcomes they had on people's lives, whether it was taking away their ability to exercise or whatnot. There were equivalent size lines at the LCBO. Very true, very true. Yeah, those were absolutely wild when quarantine first kicked off. It was just wild to see what was getting the most lineups. I mean, yeah, it was a very strange time. I guess maybe like someday we'll feel nostalgic about that. The brief moment when everyone was kind of in solidarity against the virus before everything got all weird the way it is now. Who knows? But I'm going to tell you this. I highly doubt that COVID helped anybody's health. Nathalie asks, does anyone know if we've ever seen a trend of obesity decrease in Canada's history or another part of the world? Well, the rate of gain has leveled off somewhat. So in the really rich developed countries, we're not seeing a skyrocketing gain the way we were, and childhood obesity is similarly the rate of increase has leveled off for most communities for many communities, I should say, in most of these countries. So there is a bit of a leveling off in the gain. It hasn't decreased appreciably in recent years, and I guess like the biggest contributors to a decrease would be things like the Great Depression, for example. Lots of people lost a ton of weight during that. Places where there's actual famines and food shortages and like severe bad poverty, that's you would see it then. I think it's very tough to imagine a scenario where the rate of obesity would go down markedly in a population, because it would require, I think, a pretty sizable change in the relationship between that population and its food supply. The food supply is very abundant. Food is, increasingly expensive, but it's still very abundant. Nobody's out there. There's no shortage of calories to be had. And I don't know how well we would cope as a society if our food supplies were constrained in a way that would actually make us lose weight systemically. I don't think we would react to that very well. We're a very, I think whatever. Well, nobody should have to go through that. Frankly, is the kind of restrictions needed to like massively lower weight across the entire population would be. Probably destroy society, to be quite honest. I don't think we're really able to cope with that type of thing. So I haven't seen a decrease anywhere really outside of a few cases, but it's at least the rate of gain has slowed down somewhat. Anyway, so what I was saying was that we can look at these genes one by one and kind of name them to say whether they were expressed in the brain or not. But we can also do this kind of statistically and just ask on a large scale using methods that are sort of similar to the gene ontology approaches that you would have learned in your second assignment. Just, well, where are the genes expressed? And the genes that are associated with obesity and BMI are overwhelmingly expressed in the nervous system, the brain, spinal cord, that kind of thing. They're not expressed cardiovascularly, digestive, or in the endocrine system, surprisingly, or in the immune system, or in the skin, or in the musculoskeletal, or respiratory, or stomatogenic, or urogenital. It's all brain stuff. And actually, if you look at the gene ontology categories, you see things like NMDA receptors and GRIN2A protein complexes and synaptic vesicles and nerve projections and axon termini and all kinds of other things that are very clearly brain related. So it seems to be the case that brain-related genes are the main thing that are being affected by BMI-associated variants. Just to go back to the chat for a second. So yeah, it is an interesting question. I agree as well. I think it's very interesting to consider because we're sort of working in at least I am working somewhat in the field of energy balance and obesity. And ostensibly, the goal of doing this is to try to find a way to control the rates of obesity because we're interested in the population health and we want people to be healthier and live happier lives and so on and so forth. But it's very tough to imagine what exactly we're proposing to do. As I said, one way to lower the obesity rates would be to massively increase the price of food, making it very difficult for people to get enough calories for them to survive. That's unthinkably evil to do. There's no possible scenario where anyone could ever advocate that. And I don't even know if it would even really be possible given the way agriculture is set up now. We've sort of offloaded all of the environmental externalities of agriculture, as well as the labor externalities by our use of cheap labor, to the point where I think it's very tough to imagine food prices ever skyrocketing again. Maybe at some point they'll have us eating bugs or like crickets or something to keep the calorie levels up. But it's very tough to imagine what kind of scenario could ever do that. And then what other alternative is there? Are we suggesting that we should all be taking a pill that changes our brain around to make us less hungry? What is the implication here? I think my own perspective is that the risk of obesity and your actual kind of tendency toward energy balance homeostasis is set during development. So it's not really so much a matter of taking people who are already alive on Earth today and changing their weight. I think that's basically not possible. It's more a matter of ensuring the health of the next generation to make sure that they develop in a way that sort of predisposes them toward a more favorable energy balance phenotype, we'll say. Yeah, accessibility to food is very easy these days and doesn't help things very much. But it's hard to say. I don't know. I mean, people have come up with lots of schemes to try to get into the environment and change things. I know in the U.S., the SNAP and EBT programs try to discourage buying unhealthy food by making it so that things like pop and potato chips and whatnot aren't covered by benefits. But that still doesn't help because you can always figure out a way to buy these things. You can, and I mean, I know that I think maybe New York State tried out soda taxes for a while or maybe it was New York City with Mike Bloomberg as mayor. But again, it really doesn't matter. Like the demand for this is very elastic. People will always come back for it. They'll always sort of find a way to get more calories than they need. There are so many calories in the environment that it's next to impossible to make any kind of realistic economic incentive for people to lower their calorie intake, considering that, millions of years of evolution drive us to be hungry for more and more calories all the time. Okay, I'm just going to take a quick break, folks, and I'll be back with you in just a minute. And we're back. So we've learned, I guess, that obesity genes tend to be ones that have to do with the brain. Let's look at the effect size of BMI SNPs. Because while we know that obesity seems to be highly heritable and thus presumably highly genetic, the SNPs we found don't tend to explain a whole lot. So the first BMI-associated SNP was found in the FTO gene, as I was saying. This is a very, very powerful allele. It has a very large effect size. So even the earliest GWAS studies that only had 5,000 people in them or so, they were able to detect it. And the effect size works out like this. For every copy of the BMI increasing allele of FTO, the minor allele frequency being between 40 to 45% in Europeans, your body weight, your BMI is increased by about 0.35. This is the equivalent of about 1 extra kilogram of weight for somebody who's 1.7 meters tall. And if you have a brain like mine, the only way you could ever understand this is by saying it's the equivalent of about 2.2 pounds for a 5'7" person. So that's actually not a huge amount. I mean, you could gain or lose 2.2 pounds over the course of a year just by seasonal variation, say, or just by, having a spell of depression and spending more time on the couch. Like, it's not a huge amount of difference between people with the high risk allele for FTO and the low risk allele. So, and this is the most powerful SNP we have. So the SNPs we found beyond this, they are statistically robust. They replicate quite readily, but the effect sizes are very, very small. So some of these are rare variants with minor allele frequencies as low as 1.6%. Meaning that on average, out of a group of Europeans here, only 1.6% of them would have that allele or be homozygous for that allele. They have microscopic effects on BMI, 0.04 kilograms per meter squared, it's 0.04 BMI units. That's like nothing. That's like, it's like literally several grams of weight. It's impressive, I think, that these studies are able to detect this. Imagine being able to detect genetic influences that change your weight by 40 grams. That's an astonishing amount of precision for these studies. But yet, that's like a microscopic difference. If you just look out across the world, you can see people who differ by tens of kilograms in their body weight. And yet the genetics we have maybe can only explain a few kilograms of difference between any person with giant genetic studies that look at hundreds of thousands of people. The latest studies, they encompass over almost 800,000 individuals, and they found over 750 BMI-associated variants, yet when you put them all together, they explain just 6% of the inter-individual variation in BMI. So, to kind of make this maybe a bit more visual, imagine you have, a really skinny person and a really, a larger person that, who are visibly different, and you say, okay, well, you're different by 100 pounds. I can explain where 6 of those pounds come from. That's, that's not really mathematical, but it's that kind of thing. It's like almost all of the difference in body weight between the biggest and smallest people is a complete mystery to us, despite the fact that we're pretty convinced that BMI is a fairly genetic condition or a fairly genetic phenotype. So what's the deal with that? Where is the causality? We have truly massive GWAS studies, which I should say is possible because BMI is a quantitative trait. We just talked about ASD in the last lecture. ASD is not a quantitative trait. I mean, it actually probably could be if you wanted it to be, because this autism is a spectrum, so you could actually do it as a quantitative trait. But typically it's not. People are dichotomized into either being diagnosed, having ASD or not having ASD. And there you might be lucky to get a few tens of thousands of people. But BMI, everyone has a BMI. So you can just literally go to a bus station, pick everyone off the platform and say, you're all part of the study now because you all have a BMI we can measure. We'll take your DNA, we'll measure it, and we'll see what correlates with BMI. So you have the ability to design massively powerful studies, and BMI is has a very strong genetic influence, we think, and yet the genetics of BMI still remain quite mysterious. Now, we've learned a few of the ways that GWAS studies can be limiting. There could be rare variants that we're not detecting because our SNP arrays don't see them. There could also be de novo mutations or repeat expansions or indels, insertions or deletions, or other kinds of structural changes that we're not able to detect. That is certainly possible, and maybe someday whole genome sequencing will answer this question for us, but that's still kind of out of reach, especially in studies this big as the GWAS ones. So let's change our perspective a little bit and ask about epigenetics. Because so epigenetics can sometimes, epigenetic effects, I should say, can sometimes masquerade as heritability. This is especially true for transgenerational epigenetic effects, where, say, for example, mom is obese and her child becomes obese by transgenerational inheritance. That would have the appearance of genetic inheritance, but it's actually not genetic, it's epigenetic. So this is kind of an attractive answer, actually, because as I've said, the rates of obesity appear to be rising, although the rise has slowed down somewhat in recent years. But if obesity is genetic, if BMI is genetic, it's very tough to explain why it's going up, because the genes that we have are the same as they've been for tens of thousands of years. The human gene pool cannot change rapidly over a few decades. It requires many, many millennia for the overall genetic landscape of humanity to change enough to support such a huge difference in phenotype. So genetics on their own can't explain why the rates are rising. So let's take a look at some questions of development. And I'm going to give you sort of a vignette here. I don't have time to, I actually changed this. I was going to make this a 2-part lecture, but I decided to only make it 1 part because I think I wanted to talk about neurodegenerative diseases instead. But anyway, I'll give you some things to think about, and then you can take this and kind of reflect on it on your own time. Body weight can be programmed by early life experience. This is not a genetic effect. It is something to do with brain development and epigenetics. So this was discovered in rats, at least in the 1960s, by Widdowson and McCants. They were a pair of British physiologists, I guess, and they discovered that if you breed rats and then put them, rats normally have litters between 8 to 10 pups. And if you leave them at 8 to 10 pups, that would be considered a large litter. And if you cull the litter down to, say, 4 pups, that would be considered a small litter. And you can do this because the rats are lab animals and you can just breed them and take the pups away or mix and match the pups however you like. And it turns out that if a rat is raised in a small litter, the pups get a considerably higher amount of milk from mom. The pups will take as much milk as they can get. And because mom is feeding fewer pups, she can give more milk to each pup. As a consequence, these pups grow more rapidly. But what's interesting is that not only do they grow more rapidly during suckling, which is only the first 21 days of a rat's life, they also gain more weight and remain heavier their entire life. So here is a chart of the large versus small litter body weight over their lifespan. The y-axis is weight in grams and the x-axis is weeks. The period of suckling happens only for the first 20 some odd days. So at the arrow that they've drawn here, that's when the rats are weaned. and their body weight continues to increase as they feed themselves independently. And you can see the small litter rats with the hollow circles here are considerably heavier on average than the regular size litter rats here in black. This is a huge difference. You're talking about a 33% increase in weight just by being in a large versus small litter. And this is a permanent change. You can put these rats on a diet all you like, they'll just bounce back. Now, compare and contrast this to restricting food later on. Here, what they did, it's the same sort of setup on their chart. They took rats raised in regular size litters and tried to restrict their food after between 3 to 6 weeks. So if you take a regular rat litter and restrict their food by cutting down the amount of food they have every day, their rate of growth will gradually slow. So you can see here the rats that were food restricted gradually, for those 3 weeks they don't gain very much weight, while their conspecifics are gaining considerably. But as soon as you restore the food, they go right back to their normal trajectory, and they're barely any different. They're a little bit different, but they're barely any different. So a food restriction, even a few weeks after weaning, doesn't have nearly as strong an effect as it does during suckling. And then even worse, if you restrict their food from 9 to 12 weeks, here you put the rats on a bit of a diet, their rate of growth shrinks. They actually lose weight, as you would if you were on a diet. But as soon as you give them their food back, they immediately go back onto the same trajectory and, in fact, somewhat overshoot the target or overshoot the control group. So what we're seeing here is that if you change the amount of nutrition a rat gets during suckling, the first 3 weeks of life, you get this permanent long-term difference in their body weight. But if you change the amount of food they get later on, after they're weaned, either early 3 to 6 weeks of life or 9 to 12 weeks of life, you get a temporary change, but it immediately pretty much goes back to where it was. Doesn't this remind you of dieting? I mean, isn't this exactly what it's like to diet as a person? You can diet for as long as you like, but as soon as you give up the restrictive control over your eating, your body weight bounces back to where it was. There's a critical period. If you change your nutrition early in life, it has a long-lasting, if not permanent effect. But if you change it later in life, it's too late. Your body weight is already, your energy balance is already programmed in. And Ravelli et al. Found the same thing when they looked at men who were exposed to famine in the Dutch hunger winter. I mentioned the Dutch hunger winter in our talk of developmental programming. So here, they're looking at cohorts, A, A2, B1, B2. These are all just different birth cohorts that were gestated in different intervals, flanking the famine. So the Dutch famine happened here in the winter of 1945. That's given by this section. And the individual cohorts were here. So each of these lines is 9 months. So you could have been, conceived here and been born here and so forth. And so anybody who was conceived in D1, or anyone who's part of the D1 or the D2 cohort, spent some portion of their gestation in the famine. And you can see here, this is a plot of calorie intake in the Netherlands. You can see it goes down considerably during the famine. So some cohorts were conceived and born prior to the famine. Other cohorts were conceived and born after the famine. And then a small subset of birth cohorts were conceived and born during the famine. And if you look at the rates of obesity among these different groups, what you will see is that the D1 and D2 cohorts, it's the D1 primarily, who were conceived and born during the famine, have considerably higher rates of obesity. Their rate of obesity is about twofold higher than the people who were born who weren't exposed to the famine. So I think this is a fairly dramatic finding that exposure to adverse conditions early in life can dramatically increase your rate of obesity. And it should be said that both the example in rats that I showed you previously and Ravelli's example here in humans, the change in body weight or the change in obesity risk is huge. 33% increase in body weight, twofold increase rate in obesity. Those are much, much bigger than anything we've ever found in genetics. Genetics, as I said, can account for about 6% of the variation, maybe a few kilograms of difference, but not a twofold increase in the rate of obesity. So I think this shows that early life environment has a very, very strong effect on body weight, maybe more so than genetics alone. Now, this happens in the context of the hypothalamic energy balance circuitry, which I'll tell you about just briefly if you're not familiar with it. It's centered in the hypothalamus, and it goes through a couple of developmental milestones. In a mouse, which is where most of this stuff is studied, in between embryonic days 10 and 16, you have neurogenesis, where neurons in the arcuate nucleus, which is at the base of the brain, and the paraventricular nucleus, which is toward the top of the third ventricle, are being created. And then, under the influence of leptin, which is the hormone we learned about, you have circuit formation that happens postnatally. So these neurons send axons up to the paraventricular nucleus, to these pre-autonomic neurons that live in the paraventricular hypothalamus. And then those will orchestrate all of the downstream autonomic and hormonal events that happen. So these neurons in the arcuate nucleus are very central because they are the ones that are able to sense levels of leptin, they're able to sense levels of ghrelin, insulin, blood glucose, all kinds of other hormonal and nutrient signals. They're able to detect it because of where they're located. They're sort of at a window for the blood-brain barrier. They can detect these levels of hormones and sort of make calculations about how hungry you need to be, and send that information downstream to places like the paraventricular hypothalamus that coordinates neuroendocrine and pre-autonomic functions. So the development of this circuitry happens during a very specific temporal window that happens to exactly coincide with this critical window for body weight programming. And really what's most important, as far as we can tell, is the successful growth of these axons from the neurons in the arcuate nucleus to the neurons in the paraventricular hypothalamus and elsewhere. Like I said, this happens over a very limited developmental window, and it can be upset. So if you look at these circuits in models of developmental programming, you'll see that in mice lacking leptin, the density of these fibers coming from the arcuate nucleus into the paraventricular hypothalamus is reduced. Here it is in a control animal, and here it is in an OB mouse, and you can see the density of these fibers is markedly reduced. This means that a mouse that doesn't have leptin, their energy-bound circuitry is miswired from the start. So even if you give them leptin, they're not going to be fully healed. They'll be, improved considerably, but their energy-bound circuitry will not be the same as it could have been had they had leptin in the first place. And the same sort of deficit in fiber growth happens in mice that were exposed to maternal obesity. So here we have the fiber density in offspring of obese mouse moms, I should say, dams as they would be called. And then here are the offspring of these are the controls. And then here are the offspring of the obese moms. You can see that the density of these AGRP fibers, AGRP is one of the main neuron types, are lower. So the growth of these projections that seem to be so essential in controlling our central hypothalamic energy balance circuitry is disrupted by both hormonal deficiencies as well as adverse early life nutrition. Whether it's maternal obesity or maternal food deprivation, a lot of things can do this. But the point is that this brain circuitry controls our feeding and our energy balance, and you only get a brief window of time when it's being developed. If it develops the wrong way because something's gone wrong with your early life, then your brain's ability to control your food intake and to control your energy expenditure is permanently disrupted. It's not going to be the way it should have been, and thus your risk of obesity, or your risk of having high BMI or being overweight, is permanently going to be elevated as a consequence. So we can look briefly at the epigenetics here, and I don't want to burden you too much with this because, well, for one thing, I'm publishing a study on it soon, hopefully, so you'll learn a lot more about that when my work is published. And at the moment we don't really know a whole lot, but someday we will. But we do know that in the hypothalamus, neurons do experience considerable development of their DNA methylation. So here's a volcano plot showing gains in methylation from postnatal day 0, that's the day a mouse is born, to postnatal day 21, that's the day they're weaned, in neurons. And what you see is that most CPGs gain methylation in the postnatal hypothalamus of the mouse. Okay, so that's not too surprising. Gaining methylation is a normal part of development as far as we can tell. If you look at the gene ontology terms of genes where there is this gain of methylation, the terms you see are kind of telling. You have things like cell migration, you have axonogenesis, you have growth factor signaling, chemotaxis, and axonogenesis again. Now, as I hinted to you earlier, one of the big processes that's happening in this postnatal time period is the growth of axons from neurons in the arcuate nucleus to downstream neurons in places like the paraventricular nucleus. If this growth process, as far as we can tell, is governed in part by epigenetics, including DNA methylation. DNA methylation, it seems, turns off the genes that are involved in neurodevelopment, meaning that after this methylation level at these genes has increased beyond a certain point, neurodevelopment comes to an end, and this period of early life plasticity ends as well. So you all made volcano plots in your second assignment, so I hope that the meaning of this plot is clear. In case it's not, the fold change in methylation is given on the x-axis. So anything to the right-hand side is a gain in methylation, and anything to the left-hand side of the origin is a loss in methylation. And the statistical significance, the negative log 10p, is given on the y-axis. So the higher the point is, the more significant. So points that cross the sort of a significance threshold and also are significantly different from 0, or from 1, I should say, those are significant gains in methylation. You can make volcano plots not only out of gene expression data, but also out of methylation data. And this fits in, I should say, with maybe a broader literature on the organizational versus activational effects of hormones. During early life, hormones do one thing. They change the way your brain develops and permanently establish sex differences, for example. And then later in life, they don't do that. They just activate brain circuits, causing certain behaviors to take place. So there's early life plasticity and then later life lack of plasticity. We also see age-related changes in specific genes in the hypothalamus. So for example, here is the mouse hypothalamus looking at the NPY gene and the POMC gene. These are two genes that are heavily involved in the energy balance circuitry. If you haven't heard of them, don't worry about it. It's not part of the, we're not going to be testing on that. But you see that if you look at P12 methylation levels in P12 neurons versus P35 neurons, you'll see that in certain key places, methylation levels increase significantly from P12 to P35. And again, this is a time period that straddles this critical window for neurodevelopment. You also see it here and here in the POMC gene. So there are regulatory differences that exist in these regions that seem to play a role in regulating the expression or regulating the expression of these genes in mouse hypothalamic neurons. So I want to just summarize with a little kind of diagram of the question of critical windows. What I'm trying to say here is that genetics certainly plays a role in your BMI and your risk of obesity. However, genetics can't explain everything. It especially can't explain the gain or the increasing rates of obesity that we see across the population. What can explain it, at least potentially, is developmental programming. If you're exposed to adverse nutritional conditions, whether it's maternal obesity or famine or overnutrition because your mom fed you, Coca-Cola while you were a baby or something like that, whatever, a whole variety of conditions, all up to and including socioeconomic differences, I should say. If your mom was stressed or didn't have time to give you the proper amount of nutrition, or if, in the States, maybe you don't see it. I actually see it here too. In a lot of places now, baby formula is literally kept under lock and key at the store. There's a lot of sort of perverted conditions out there in the world. But anyway, there's all kinds of reasons why things may not be optimal for you during early life. However, early life is a critical window such that if bad things are happening to you nutritionally or otherwise, it can have a permanent effect on the way your hypothalamic feeding circuitry develops. So here is the critical period in mice. it goes from about P0 when they're born to around P28 after they're weaned. Over the course of this, you have a spike in the expression of the de novo DNA methyltransferase, DNMT3A. You have a gradual increase in the projections, the axons going from the arcuate nucleus to the paraventricular nucleus, as well as other neurodevelopmental events. And at the same time, you have an increase in DNA methylation in neurons. And all of these events are happening at the same, across the same window of time, creating a process that is very vulnerable to disruption. If something were to happen here that changed the way the DNA methylation was being established, you could have permanent differences in DNA methylation in your brain that might permanently affect the way your energy balance genes are regulated. DNA methylation seems to regulate the process of neurodevelopment, meaning that the reason why neurodevelopment is fixed after this point is because DNA methylation on neurodevelopmental genes has reached a stage where it can no longer be changed by the environment. It's kind of stamped everything in, everything is fixed, all the screws are tightened, you've put the access panel back over the mechanics, and you're said, we're done. This is what happens after the critical period is over. So if you want to kind of summarize this, there are a lot of critical developmental processes happening during early life. Many of them are involving epigenetics. And because because so many things are happening, there's a much higher level of sensitivity to disruption by the environment. And disruption that happens here has the potential to be permanent, if not very long lasting, meaning that it's very, very difficult to change later on by diet or by exercise or by what have you. So what is obesity? It's a medical term applied to individuals above an arbitrary cutoff on an arbitrary scale. And I feel compelled to mention this because I think for all the amount that I talk about obesity, I don't really like the way that the research or the way it's framed can be sort of stigmatizing to people. It doesn't help, especially now that I've told you that, at least in my view, it's not something that can be changed very easily. And if it can't be changed very easily, there's no use in moralizing about it and making people feel bad for the way their bodies happen to be. Much better, I think, to focus on actual health and focus on trying to help people live the healthiest lives they can. That's a separate question, of course, from how should we manage development. If there are problems with the environment that are causing people to be less healthy, then we should certainly address those. And that would mean changing the way, changing many, many aspects of how we treat young families and mothers and people who are vulnerable or people who are in a vulnerable state in their life. It's determined, obesity, I should say, is determined in large part by genetics and early life exposure. But once the individual's obesity or sort of energy balance set point is established by a combination of genetics and the environment, it's very refractory to later changes. As you know, diets are just not very effective on the large part because they do not produce permanent changes in your brain. The energy balance circuitry really only changes during early development, and once it's set in place, it's going to be, it's not exactly possible to change. I think about this myself as somebody who's very skinny. It takes no effort at all for me to be skinny. It happens automatically. But if somebody were, say 20, 30 pounds overweight and dieted very carefully to get down to the body weight that I'm at, they would not be the same as me. They would be living in a very different world where their brain would be continuously reminding them how hungry they are. It'd be like trying to work with a fire alarm going off all the time. Even if your bodies sort of physically look similar, the underlying energy balance circuitry could be quite different, meaning that somebody like me who happens to have just been born as a skinny person, it's going to be a very different situation from somebody who was born to be a larger person and worked very, very hard to get down to being skinny. This is a fact of life, and I think it has to be acknowledged when we're discussing about, when we're discussing what to be done about the so-called obesity epidemic, bearing in mind that it's like largely an epidemic of brain development that's gone a certain way and programmed people to have a certain energy balance set point. So if you take home one lesson from today, take home my contention that obesity is a neurodevelopmental phenotype. The genes that are linked to obesity and BMI are overwhelmingly brain expressed and neurodevelopmental in character. Almost all of the genes are brain related. They're not fat related, they're not stomach related, they're not muscle related, they're brain related. And the part of the body that is most vulnerable to the epigenetic and well, neurodevelopmental effects of developmental programming, which, as I've shown you, have a very strong effect on obesity risk. That's all in the brain. It's all happening in the brain, folks, and most especially in the hypothalamic energy balance circuitry. So this is what obesity is. And I think what ultimately it contributes to, or what your energy balance set point contributes to, is a natural tendency toward finding a set point for your body weight. Your brain has, in a sense, a body weight that is ideal for you that you will always try to get back to. If you limit the food that you take in, you can deviate from it temporarily, but typically your brain is going to try to defend that body weight with whatever it can. Such that if you're on diet, typically the first thing that happens is your brain shuts down or lowers your energy expenditure so you can conserve energy and lose weight more slowly. So I thought maybe it's a little bit like, a good analogy for it would be like the Stroop effect. If you remember from your psychology classes, the Stroop effect is when you have to, it's like you have these words these words spell out certain colors, but then the words themselves are colored. And I think the idea of the Stroop task is you would come down here and you're not supposed to read, you're supposed to say what color the letters are, not what the word says. So it's very tough to overcome your brain's natural tendency to read these letters. So you say okay, so this is yellow, red, blue, green, purple. It's very tough to overcome your brain's natural tendency to read things. And so people don't perform well, even though it's a comparatively simple task. And I would analogize dieting to the same thing. It's very simple to eat less food. However, it goes against everything your brain is telling your body to do and all of your evolutionary imperatives, as well as all of your developmental programming and everything else. So not an easy situation. I think very important to bear in mind when we're talking about this. So there you go. That's not to say, of course, that obesity is a mental disorder or a mental illness or anything along those lines. It's just an aspect of brain development that can be affected by the environment and by genes. And really, if you want to zoom out and look at a bigger picture, that is actually the way we would describe most conditions that we're talking about. They're neurobiologically rooted phenotypes that can be affected by genes and by the environment. And so by studying obesity, which happens to be a fairly popular model and fairly well-funded, comparatively speaking, you can learn a lot about the way genes and the genetics and the epigenetics contribute to disease risk. All right, my friends, so we'll leave it there. Hopefully you're having a good week so far. Hopefully things can only improve for you. We have office hours tomorrow if you want to stop by. Otherwise, have a great weekend, my friends, and we'll have more to say coming up on Monday. Please enjoy the last third of your semester here. The end is in sight. You just have to kind of push it a little bit longer, and you'll be home free for the holidays at least. So enjoy, and we'll see you next time.